right? But the summers in Boston are pretty fantastic. They are pretty fantastic.
Yeah. We can't complain here. All right, it's 4 or 5 p.m.
Oh, wow.
And I think that's sort of our official time to start. I think there's still people trickling in. I'll make a couple of announcements. We have these Meki colloquiums going for the next couple of weeks. And next week we have our own Professor Stephanie Müller from ECS and Mechanical Engineering, and she speaks about personal fabrication using sort of dynamically changeable everyday objects, if you want. That's going to be exciting. But you're here today for speaking or hearing Professor Manu Platt speak. And one thing I want to get out of the way before we give this to Manu is, if you have questions, put them in the chat. If you want to be on the panel, I would be very happy to promote you. Just write something in the chat. I want to be on the panel, and I'll put you up here so that Manu can look at friendly faces. And then you would have the benefit of asking questions very quickly if you wanted to. The other thing I should do is give you a bit of an overview of how cool Manu is. I've been sort of stalking him and his website a couple of days now to get a bit of a feeling for what he's been doing. And the work is truly fantastic because he does this thing where he takes biomechanics and biochemistry and bioengineering in the context of tissue mechanics and tissue sort of remodeling. And then he sort of solves really tractable and really important disease problems, either in the context of the children getting strokes because of sickle cell disease or breast cancer and personalized medicine, or HIV related sort of complications. I think cardiovascular problems is the word I should use here. So the work is tremendous, and I have a few slides to share with you on that, because I want to show you some of this stuff, and then he can show it himself. Here is a slide that I pilfered from his website. So here's the announcement. You have seen that before, but what I want to show you is sort of a slide here. His work goes into global translational research in terms of helping with disease states, and that not only in the US, but also in Africa, South Africa and Ethiopia. He studies systems biology and medicine, but he's also a member of the EBICS Center, the Emergent Behavior of Integrated Cellular Systems that Roger Kamm is leading, together with Mary Griffith and other people at MIT, Georgia Tech, I think at USC as well, and a couple of other places. And he uses modeling and experimentation alike. So that's really cool. It's a really sort of complete package. The other cool thing is something that is best conveyed by this picture here. So I stole this from Manu's website. That shows his lab 2 years ago at a 10-year surprise reunion. And there's no single person in this picture that doesn't smile. And I think that gives you an idea of how influential Manu is in terms of influencing people in science, but also in life. And that's a cool thing. And that is actually a point I want to drive home with one more thing. Manu cares a lot about sort of the environment, not only the health of you and people in general, but also the health of the scientific environment. And you can see a really cool sort of engagement, or if you want, initiative here, a program that I think exists since 2013, and that's Engage. Manu and Bob Naran fund this at Georgia Tech and Emory University, and you can see here a couple of high school students, aspiring scientists from all sorts of different background in the sort of minority, serving public high schools, that Manu brings to science and to companies with Bob and other people that help. So that's really sort of the level of impact where you say, this is awesome, you have to talk about this. And so now, Manu, with all this excitement, I want to hand this over to you, and everybody wants to listen to you.
Wow. Wow, thank you. Wow. You know, you come into these things with your mind like ready to do one thing, and wow, you just got to flip my mind a little bit. Thank you very much. Those are great pictures. Thank you for that introduction, and thank you all for having me. Again, thanks to Matthias and to Ming for the invitation, and I think Roger Kim had a little something to do with it, plus my old wonderful mentor, Linda Griffith. So again, today I will be talking about, again, from the topic about sickle cell disease. I think about it in the context of strokes for the children. But there is this dynamic interplay between biomechanics and biochemistry. And these are things that I've been studying ever since I started grad school. That really is what brought me into biomedical engineering, because my undergrad degree is actually in biology from Morehouse College, but I love biomedical engineering because it's about how does the physical world apply to the body. And our bodies and cells have to obey those same principles as a brick or a feather that drops. So greetings from Atlanta. It is wonderfully warm down here, but I hear it's warm in Boston. So you all are getting really lucky so far. But okay, let's kick it off. Thank you. So I run the Platt Lab for Repair, Regeneration, and Remodeling. We were established in 2009. But our big areas are cardiovascular tissue remodeling. I'll talk a lot today, clearly from the title, about our work in sickle cell disease. We've done some work with HIV and cardiovascular disease, which really took me overseas quite a bit. I'm happy to be a part of the center EBICS, Emergent Behavior of Integrated Cellular Systems. As mentioned, some really great connections and interesting ways of thinking about using biology and cellular processes. And then my work on systems biology and predictive medicine. This was really spawned out of the postdoctoral work that I did up at MIT with Linda Griffith and Doug Laufenberger, where I really wanted to learn more about computational modeling and applying it to biological systems. So check us out, plattlab.com. But I say I run a lab on repair and remodeling. So I like to tell people, what do I mean when I say tissue remodeling? So I think about it as how do you go from this healthy tissue to this diseased or damaged tissue? So I'm actually gonna turn on my laser pointer, there we go. So if you think about this is a human coronary artery, a healthier one, that it's open where blood would be coming out of the board and be able to pass through the coronary artery. Corona, that's been sneaky again. But then how do you go from this healthy artery to this plaqued vessel that has this huge plaque that has now been formed? Here's where the lumen actually was of that blood vessel. So you can imagine it can no longer allow blood to pass, and because it's a coronary artery, this would be a heart attack, right? So some of the initial steps of this tissue remodeling are protein degradation of some of the key structural proteins that make up the artery wall. Then, of course, new proteins are synthesized and laid down, and of course, cell infiltration happen to remodel this tissue. And so my lab wants to understand what are these mechanisms so that then we can stop them or identify pharmaceutical targets for therapy. Most people know about osteoporosis. You go from this healthy mineralized bone matrix with a lot of type 1 collagen that's been mineralized for strength. But then you get an osteoporosis happens predominantly in postmenopausal women, where you get degradation of the collagen 1 and demineralization. And again, not only do you lose the biomechanics of the of the bone, but you also lose some of the signaling that happens in the bone as well. I like to show this. This is an example of one of the biological machines developed during EBICS with Rashid Bashir's group at University of Illinois. And what my group's questions were how come when we've designed these biological machines, they then break in the middle? So what remodeling is happening, and then how can we understand the remodeling processes so we can then engineer functional lifetime of these biobots? But today we're talking about sickle cell disease. So there's all these engineering applications, but again, human health and disease is what kind of keeps me up, makes me want to work hard and solve problems. And I call this like the singularity of sickle cell disease, right? For all of us who were back in like AP biology back in the day, you learn about diseases that are single point mutations, usually may not be so problematic. It's the frame shifts that cause the big problem. But sickle cell disease is a single point mutation disease that causes a multifactorial problem. So I call it the singularity. What does that mean? One nucleotide is changed from an A to a T. This, of course, changes one amino acid from a glutamic acid to a valine, which is in one protein, hemoglobin. But hemoglobin is special. Hemoglobin is a protein that is only in one type of cell, red blood cells. Many of the other proteins we study are in multiple cell types. Hemoglobin is just sequestered in this one type of cell. But what's the problem with red blood cells? They go everywhere. And that's why you get this multifactorial damage of all organ systems due to sickle cell disease. And it causes the sickling of the red blood cells. A lot of people who don't hear about it or talk about it as much anymore think, oh, it's cured. We've known about it. It's been studied clinically and research-wise since 1910. And is actually, but still, the only cure is bone marrow transplants, which there's issues there we could talk about later. But the life expectancy today for men is 36 in the United States and 42 for women. And it is a health disparity in the United States. 1 in 400 African Americans have sickle cell disease, and 1 in 12 has sickle trait, meaning they are carriers or heterozygous, for those that know what that means, which means if 2 people that have sickle trait reproduce and have a child together, there's a 25% chance their child will have sickle cell disease. And many people are not aware that they have sickle trait, even though we're all tested at birth. But although there are 100,000 Americans with sickle cell disease, making it the most common inherited blood disorder in the United States, there's so many more millions worldwide. So there are 300,000 babies born each year with sickle cell disease. So this exploded cartogram kind of gives an indication of where. Sub-Saharan Africa is a major area. The Indian subcontinent has a high population of sickle cell disease, the Arabic world, and you can see in North and South America, and that's mostly come due to the international slave trade from back in the day. But here's the thing, globally, up to 90% of these children will die in their first 5 years of life. So I'm a heartstrings kind of guy. Scientific questions that pull at my heartstrings make me want to study them. And why do most of these die is usually due to infections. And so if you put a child with sickle cell disease on penicillin through the first 5 years of life, then they can fight most of those off. But again, this is a global health problem. And so I like to always remind people, especially younger scientists, the importance of being a scientist activist. And I think this is actually quite important in today's world and what we are dealing with right now. But I'm a child of the 80s and 90s. And when I was young, there were these public service announcements on TV that said, think globally, act locally, right? You can act in your local community. But the world shrunk when I was an adult. So then act globally. The world is spreading apart as we are isolated in our own country, but still, when it reopens, think globally, act locally. And why? Because global health concerns are health disparities for underrepresented minorities in the United States. And that also becomes a great way to motivate the work. If it's only a small number in the United States, it probably has a large global impact that you can consider. And again, this importance of becoming a scientist activist, because we need to communicate this to the different communities that may be marginalized, not as exposed to science or the medical community, to tell them what's happening and how to really protect themselves. So sickle cell disease, the molecular origins. Again, all these things are known. Since 1910, we started to study it clinically. We know the molecular origin, won the Nobel Prize for Linus Pauling several years ago, obviously. But so what happens is when that glutamic acid in hemoglobin, it's mutated into a valine. So valine, of course, is a hydrophobic amino acid, glutamic acid being hydrophilic. Hemoglobin undergoes a conformational change when it gives off oxygen. And under that conformational change, that valine is now exposed on the surface of the hemoglobin molecule. That, of course, is not thermodynamically favorable. So those hemoglobin molecules self-assemble to sequester the hydrophobic valines. And it's a very highly ordered polymer that forms, that completely deforms and distends the red blood cell membrane. The other thing to remember about red blood cells, they do not have a nucleus. So we know that nuclei are usually the most rigid body part of a cell. So without a nucleus, that red cell is completely free to be deformed by this sickle polymer. So we took this video in our lab, and you can see there are some cells that are actually more responsive to the sickle polymer. Others are more stuck in place. And we'll talk about why that's happening momentarily. So as the red blood cells sickle, that membrane continues to be damaged over and over again. So think, under deoxygenated conditions, they give off oxygen, sickle polymer forms, red cell sickles, and then when they make it back to oxygenated circulation, the polymer melts and the red blood cell is again deformable and can move again. So these are some great electron microscopy images of as the red blood cell sickles and continues during its lifetime. Again, under oxygenated conditions, these discocytes here look more like a wild type red blood cell with the biconcave disc and that freely sickle. But as the cells continue to sickle and they age, their membrane gets more viscous, the damage accumulates, and not only does the membrane get more, so the membrane gets more viscous until ultimately, whether it has sickle polymer inside or not, the membrane has retained this shape and damage because of the viscosity. And at this point, these irreversibly sickled cells can then break open and lyse. And that's why you also hear about this as sickle cell anemia, where they have less number of red blood cells than a person without sickle cell. But the other problem for these individuals is they have what they call the pain crisis or a sickle crisis. You can see these spiculated sickled red blood cells. They give off oxygen in the capillaries, and then in the post-capillary venules, they can get stuck, adhere to the blood vessel wall, and start to block blood flow. And this is a huge sickle crisis. This is actually happening all the time for people with sickle cell, but when it gets to be a large enough accumulation and a large enough pain, that's when it's called a sickle crisis and they're rushed to the hospital. And how do we treat a sickle crisis? We give them saline and wait. Painkillers and saline, and that is the current medical standard today. But someone did some studies years ago about sickle cell while she was a graduate student. This is Linda Griffith's paper when she was a grad student at Berkeley, because a lot of people who study sickle cell actually studied in the microcirculation for these reasons. When the red blood cells give off the oxygen, they begin to deform and get stuck into the microcirculation. And she actually has some really nice mathematical studies here showing that there's a critical viscosity of the plasma that can lead to this clumping and sickle crisis of the red blood cells. There she goes there. And so again, what's different from a normal red blood cell is as a person without sickle, as the red blood cells normally move through capillaries, the velocity of the red blood cell as it enters the capillary is the same as it comes out. And same thing for the viscosity. But in a person with sickle cell, again, as their red blood cells are giving off oxygen, the viscosity of the blood is increasing as it is of the red blood cell, and their velocity passing through the capillary slows down, which allows them to give off more and more oxygen and to sickle more and more. So this is just my updated figure from Linda's figure from back in the day. But there's some other interesting things that happen here. So in a person without sickle, again, about 45% hematocrit. This is the percent of red blood cells per unit volume. All right. Sickle cell is known as an anemia because they only have about 25% hematocrit. So way less red blood cells per unit volume. But here's the real importance of this. This actually works out very well for the rheology of the blood because there's the same rheology or viscosity of the whole blood of a person with sickle as there is for a wild type person with 20% more red blood cells per unit volume. This becomes a problem clinically if a person is rushed to the hospital for a crisis and the doctors are not used to dealing with sickle cell. They may run a CBC and say, oh, they're anemic. Let's give them let's give them blood. But that would clot them if they gave them wild type blood, because if they had an equal amount of hematocrit, you would get sludge flow and cause massive clots. But what started to really get me is not the microcirculation, it's that children with sickle cell disease were suffering from strokes. 11% of children will have a major stroke. This is a paralytic stroke by age 20, but up to 33% will have what we call a silent stroke. This is a stroke in the cognitive region and the cognitive part of the brain, so a thinking part. A paralytic stroke, you might see that there is impaired motion, but in the thinking part, you got a little black kid sitting in class getting bad grades and they're saying, oh, he's stupid. And then if they look in the MRI, oh my goodness, this kid was having strokes. So this is actually the brain of a 6-year-old girl. This is a MRA, magnetic resonance angiogram. And you see this whole part of her brain is not getting oxygenated blood. So this is a major problem. And again, the greatest age risk is between the ages of 2 and 5. So imagine that. Like during my PhD, which I'll talk about in a second, we were studying people getting strokes in their 6th, 7th, or 8th decade of life. But this is happening at a much more accelerated pace. And so this really began to draw me in. And in autopsy specimens that they used to gather from the children back in the day, they no longer do it because they kind of know what's going on. They saw this massive remodeling in the cerebral arteries. So this is intimal thickening that has happened here. This is the neo-intima that has grown in the lumen of the middle cerebral artery. And this elastic lamina that provides elasticity to the arteries, you can see here it's fragmented, it's duplicated, and it's frayed. So you're losing now some of the mechanical nature that you want arteries to have for elastic recoil and other structures. And again, this has happened quite rapidly, right? Like I showed you that atherosclerotic plaque in the beginning, that was like a 60-year-old person. So there's been lots of hypotheses about why this is happening, but it has been confirmed that there is degradation of that elastic lamina by proteases or enzymes that degrade other proteins that then promotes a cellular response that can start to close off that lumen. There's a number of other things that happen that we could talk about later, but it's indicated that there is proteolytic remodeling and tissue remodeling in those arteries that's accelerated due to sickle cell disease. Where also does it get to where biomedical engineering or the mechanics become important? There was a fantastic clinical trial that was run in the early 90s by Bob Adams at Medical University of South Carolina, where they showed that if they did transcranial Doppler by measuring velocities through the cerebral arteries of young children with sickle cell, if the centerline velocities were greater than 200 centimeters per second, those children were at greater risk of having strokes. So the clinical trial was that they took children with transcranial Dopplers greater than 200 centimeters per second. One arm, they put them on monthly blood transfusions. The other arms were the placebo. And this is one of those clinical trials that ended early because it was obvious that the children getting blood transfusions were surviving while the other ones were having strokes and some were even dying. So this is one of those trials that was ended early. And the clinical standard came to be If your increased blood velocity is greater than 200 centimeters per second, monthly blood transfusions. This is still the current state of the art. This is the still standard of clinical care in sickle cell. And you can imagine all the complications with monthly blood transfusions. But so then I come into the field and I start to wonder, I took these great fluid dynamics classes when I was in graduate school. And we learned that if the center line velocities become increased, that could either be due to a stenosis. That's one possible reason. But what could cause a stenosis? Maybe accumulation or aggregation of the red blood cells or the white blood cells. As I mentioned, the red blood cells are quite sticky and adhesive in sickle cell, and they also trap white blood cells. There also could be the luminal narrowing as the neointima is growing into that lumen region. But the real complication is that downstream of that stenosis is post-stenotic flow, which causes flow separation and flow reversal, which we knew from atherosclerosis studies causes vascular remodeling to help close off those arteries. Excuse me. And so again, what does that mean? So when I first started grad school, what we were first told, given this chart from 1966, and where do you see atherosclerotic plaques forming? At branches, bifurcations, and sharp turns. And the pathologist quickly went to talk to his aerospace engineer buddy, and he said, oh, well, we know from fluids that at branches, you get regions of flow separation and flow reversal. And so that's what's happening in the arteries. And where you get these regions of flow separation and flow reversal are where you were finding atherosclerotic plaques forming inside of our artery wall. But the straight parts were protected from the plaques being formed. So this was like the first thing that happened when I was in grad school. This had been known, but I was mind blown, like, whoa, just the mechanical forces influence gene and protein expression that can cause disease or protect from disease. And so my PhD was about identifying these mechanosensitive genes and proteins that are either expressed or upregulated under the regions of disturbed flow, or those that were upregulated under the good protective flow, so that we could kind of see which proteins are atheroprotective and which are promoting the plaques. And what popped out was these wonderful enzymes called cathepsins. I'm going to say cathepsins about 25,000 more times during this presentation, so I don't want you to get sick of them. Love them the way that I do. All right, so what are they? Let's start our love affair. Well, they were first identified in lysosomes. For those that aren't like cell biologists, lysosomes are the trash compactors inside of cells. So they're responsible for degrading the other proteins to recycle the subunits, like the amino acid subunits. But they started to be identified in a lot of tissue destructive diseases like osteoporosis, atherosclerosis. We've studied them in tendinopathy, and they play a major part in cancer and metastasis of the tumor, or how it breaks out of its primary location to go throughout the body. But what I think is the most fascinating thing about them is they are the most potent mammalian collagenases and elastases we have. So what does that mean? They degrade type 1 collagen. Type 1 collagen is a protein in our bodies that has the greatest amount of tensile strength. It's 95% of the organic matrix in bone. You see those beauty commercials about increasing collagen in your face. They can degrade this better than any other enzyme we have. And also elastin, that protein I was just talking about that's in the artery wall that provides elastic recoil, they can also degrade this better than any other enzyme. And so that's where they become important. And so once it began to be realized that they were upregulated in diseases and secreted or released by cells into the extracellular space, started to then be appreciated that they can do damage and are participating in these diseases. But what's the trick? So now that once they realized they were really potent, big pharma really tried to develop small molecule inhibitors against the cysteine cathepsins. And there have been 16 cysteine cathepsin inhibitors that have made it to human clinical trials, and 16 have failed human clinical trials. We have 0 in our medicine cabinets right now. There's a lot of tricks to working with these enzymes. Oh yeah, let me put my denied. The last one was odonacotiv from Merck. It was an indication for osteoporosis. 12 years, 16,000 women in this study, 8,000 in placebo, 8,000 in the drug arm, and it actually increased bone mineralization by 67%. So a lot of times these drugs are effective in the disease, but they have not been approved because of side effects. And so there's questions now about how do we appropriately use these inhibitors and what do we not understand about the way these enzymes work that is causing the inhibitors to fail? Because I think that's billions of dollars. So that's a whole nother aspect of my work that I'm not gonna put the biochemistry part on you mechanical engineers, but it's a fascinating story. Okay. So the first thing we did is when I started my own independent lab, I wanted to improve the ways we could detect these enzymes because the assays that I had to use as a grad student required 3 or 4 ways. They were quite promiscuous because these enzymes can work on the same substrates. So just for the sake, for what you all know, we could talk through this later. We discussed developed this multiplex Cathepsin zymography, but how to follow along. Where you see a white band, that's where there's more of the active form of this enzyme. So the increased white band you see, that means there's more active enzyme. Cathepsins are named by letters. So K, L, S, V, these are 4 different cathepsin protein family members. And what we found with zymography is they all migrate to a different distance, such that if they were all running together, we could quantify each one individually. And for those of you that do cellular assays, there's no antibodies required for this, so it's a great way to save money. And this is what has allowed me to take this work into developing countries like Ethiopia and South Africa, as Matthias mentioned in the intro. But we also found it was more sensitive than Western blot, which is a common way that cell and molecular biologists quantify the amount of protein. So the zymography, we found out, was able to detect femtomole amounts of mature cathepsin K. Femtomole, that's to the negative 15. Okay, so congrats all you nano people. Come down here, see me, okay? But also that we can multiplex it and detect 4 in one assay. All right, and so we're gonna be using this throughout. But again, I also want to shout out my EBICS work with Roger Cam. This work has just been accepted for publication, where using even microvascular niche as part of EBICS, we identified that cysteine cathepsins were altered by flow. And so shout out to Simone Douglas, who was the first author. Grad student in my lab, she's now doing a postdoc with Paula Hammond at MIT right now. But I use cathepsins in many tissue destructive diseases, as I mentioned. Clearly, I've been talking to Linda, so we show that they were elevated in endometriosis, lesion establishment, cancer, tendinopathy, and the other diseases that I mentioned. But back to sickle cell. So what do we know that they do? They can degrade the elastin and actually promote plaque formation. Because when the elastin is degraded, not only do you start to mess up the elastic recoil of the artery, but there's different biochemical cues that the smooth muscle cells receive when they used to be in touch with elastin. Without elastin, smooth muscle cells start to proliferate and migrate into that plaque forming space. So what we then, all of that sickle cell ideas together, came together where we wanted to look at sickle cell in large arteries, in strokes. And remember, these large arteries are oxygenated blood, not the deoxygenated blood where we know that the red cells sickle. And so from the molecular perspective, we wanted to investigate the enzymes that were being upregulated, but then how the cells, these endothelial cells that line the blood vessels, respond to both the biochemical insults, there's chronic inflammation in sickle cell disease that activates the cells, but also the biomechanical insults and changes in blood flow that are caused by these stiffened red blood cells and with their increased viscosity as they move through the vasculature. And how together that impacts the structure of the tissue organ, we're talking about the arteries, that might lead to some biomarkers or some pharmaceutical targets that we can use. And this is the work that, and we do this multi-scale approach, and this is what my new innovator was funded on, which I was super excited about. But so we first started with, everyone wanted us to look at red blood cells, and we did look at the red cells because sickle cell is known as a hematological disease or blood disease, but we cared about how it affected the tubes, right? So we were kind of talking to the wrong people. But the red cells actually did not show much of the effect. But what we did find is that if we isolated white blood cells, these are peripheral blood mononuclear cells from people with sickle cell, they actually induce some interesting programs when they bound to the endothelial cells that line the blood vessel wall. So we actually work closely with the Sickle Cell Foundation of Georgia, community-based organization, and we actually got our donors of blood from there. They have a lab on site where they also diagnose people with sickle cell, but they also have a research arm. So Phil Keegan was the lead author on this work. Ms. Richard was the lead lab technologist. And so one of the initial things we found, again, we confirmed it in our own hands, was that people with sickle cell, SS means sickle cell. So they're homozygous recessive. So SS is sickle cell. AA means wild type. So nothing at all. And we found that if we took white blood cells, these peripheral blood mononuclear cells from people without sickle, they rarely stick to the endothelial cells. And they shouldn't, because they should just flow past the cells and your blood should keep moving. But if we pre-stimulated them with TNF alpha, which is an inflammatory cytokine, which again, this is known in atherosclerosis literature, also shown in sickle cell, you get an increased adhesion of the peripheral blood mononuclear cells. But if we isolated white blood cells from people with sickle cell without any exogenous stimulation, you can see they were much more ready to adhere, okay? This has been shown by other groups before us, but we quantified it to confirm we could, stick with the field. And you can see orders of magnitude greater number of cell adhesion in people with sickle cell. Also, because of the damage that is constantly being caused by these sickled red blood cells, chronic inflammation and damage. So there's increased circulating amounts of TNF alpha, this inflammatory cytokine that's like a danger response. And we quantify that with ELISA from the blood that we were able to obtain. But of course, what we cared about is what is the upregulation of these proteases that might cause remodeling in the artery wall. And this was quite fantastic. So from our zymogram, we see here the wild type PBMCs, you see baseline expression of cathepsin V, cathepsin L. If we pre-stimulated with TNF alpha, we see cathepsin K is now turned on. It was not on in the wild type. But with our sickle cell PBMCs, again, no exogenous stimulation. You're seeing an increased amount of cathepsin K without adding anything. So these are primed to be proteolytic. The important message about cathepsin K, it is the most powerful collagenase we have. So there's collagen in our artery walls, but it is also a very powerful elastase. So it can cleave both of those structural proteins at high rates. And it's being turned on when these sickle PBMCs are sticking to the endothelial cells. All right, and so, we're out of time. Let me keep cracking. So we looked at the cell signals involved here, and I'm going to skip through this for the sake of time, but we showed that J and K and C gene, these are cell signaling molecules that were elevated when the sickle red blood cells adhere to the endothelial cells. So to test if they were playing an important role, we again inhibited J and K with a J and K inhibitor, SP6-00125. And when doing that, either when the endothelial cells were stimulated with TNF alpha, where they turn up cathepsin K, so TNF alpha that's elevated in sickle cell, all by itself turns up this enzyme. But if you add the J and K inhibitor, you can reduce that signal by about 50%, or this is less than 50. But the sickle cell PBMCs alone turn on cathepsin K, and with J and K, you can inhibit that signal by about 50%, showing that this is a pharmaceutical target that is able to reduce the amount of the protease that's being active downstream of the sickle PBMCs or the inflammatory cytokine. So that's biochemistry. Now, where does the biomechanics come into play? Well, as I mentioned, the types of fluid shear stress on the endothelial cells influences gene and protein expression. So this is a vasoprotective profile. We pulled this. There's some other familiar names you all might know. Paper from Roger Cam and Michael Gimbroni from back in the day, where they took a physiological profile of the shear stress on the protected side of the carotid artery. And then they had a profile of the disturbed flow side, where you would promote remodeling. So we can use a cone and plate shear bioreactor that C. Forbes Dewey, another MIT professor, kind of pioneered for cardiovascular work. And we can independently program these motors to actuate these either vasoprotective or pro-remodeling shear stress profiles. And again, for those that don't know, endothelial cells are mechanoresponsive. Under vasoprotective or unidirectional shear stress, they elongate and align in the direction of flow compared to no flow. And in the pro-remodeling or the disturbed flow conditions, they do not. Okay, so it's not one way you know that your flow profiles are being actuated. And so this is like some of the really cool data where this is why I love biomedical engineering, okay? Because again, in biology, you learn like this happens, but in engineering, you can learn why it happens, and then that mechanical forces influence biology. So I told you, TNF alpha turns up cathepsin K. You see that under no flow conditions, TNF alpha vehicle, this is just endothelial cells that line the blood vessel wall. But under pro-remodeling shear stress, this is my PhD work, pro-remodeling shear stress turns up cathepsin K a little bit. And of course, TNF alpha under pro-remodeling turns it up even more. But check this out. Under the healthy blood flow, the vasoprotective vehicle, you don't see K, but even in the presence of the inflammatory cytokine, cathepsin K was not being turned up. This was crazy, right? Because the chemical is there, but the mechanical force was wiping it out. I think I meant to take this slide out. So we then looked at what was going on here, right? And so we looked at that downstream signal that I talked about, that J and K. It's an intracellular signaling molecule that gets phosphorylated. That means it's being activated. And you see when you activate it with or you stimulate the cells with TNF alpha, J and K is activated under static and pro-remodeling conditions. But even when we did the vasoprotective shear stress, where you do not see the enzyme being turned up, the J and K is still being activated, right? So we expect, our hypothesis was that it probably stops J and K activation. It did not stop that. J and K is still activated, but the protease is still not made. And so we went then, and the downstream target of J&K is C-Jun. So J&K phosphorylates C-Jun. So all of this is still happening, even though we're not seeing the active protease. So then we went and looked at the regulation at the DNA level. So we call it the transcriptional regulation. Is the gene actually being turned on under these conditions? And we found that under the vasoprotective shear stress, we were not getting the messenger RNA being made. That means the gene would be turned on. But the next question was, well, C-Jun, I'm sorry, let me go back. So those signaling mechanism that I just talked about, J&K and C-Jun actually goes into the nucleus and binds to the cathepsin K gene to turn it on. It's what we call a transcription factor. So we did a chip assay to see if the transcription factor was still going into the nucleus, binding to the DNA to turn it on. And it was. It was crazy, right? So it actually is still binding, although we are not getting the gene being expressed. The mRNA is not being made. And so I'm going to cut to the gist of the story. What we actually found out is that the biochemistry was regulated by TNF alpha signaling through J&K and C-Jun and called this AP1 transcription factor. But under vasoprotective shear stress, it actually degraded NF-kappa B as different signaling protein, and NF-kappa B was not able to go to the nucleus, and both were necessary to turn on the gene. So we have the shear regulated component and the biochemical regulated, which is why we call it this dynamic interplay between both. But this is another reason why exercise matters, right? When you exercise, your heart rate increases, you increase your vasoprotective shear stress, even in the regions that would have disturbed flow, and you end up protecting your vessels from the remodeling. So that's what we did in vitro with cells. But do we have an in vivo model? Well, let me agree. So we have a town sickle cell transgenic mouse. They knocked out the murine hemoglobins and knocked in the human hemoglobins, and then it can just be bred by Mendelian genetics, okay? And a lot of what you'll see is the AS. This is the heterozygous case, the carrier. And we do this because we can get litter mate controls for these studies. Sickle cell mice, even if you do Mendelian genetics and you breed an AS mouse with an SS mouse, you're supposed to get 50-50 being sickle cell and 50-50 being, sickle trait, but you don't. You usually get about 2/3, 1/3 because it is a deadly phenotype. So we use the AS as the gene controls. And so we wanted to know, because in young children, we were seeing this remodeling that was causing these strokes. And so in the youngest stage of these mice, we sacrificed these mice after 1 month. So that's right at weaning. And we looked in their carotid arteries and see, do we see evidence of proteolytic damage? And again, shocking. I mean, again, this is older data, but it still continues to shock us. That at that earliest age, we were seeing this increased elastic lamina damage. This is a Van Giesen stain of the carotid artery of these young mice. And you see that there's this fragmented elastin here. You can see also there's this kind of distension to the artery wall at 1 month. And we quantified the number of elastin breaks over a number of arteries, and you can see it was significant. And so then we did staining to see if we can identify the amount of cathepsin K, which is the enzyme that we shown in the human cells was upregulated. And when we used antibodies sustained for cathepsin K, we did wild type, the heterozygous, and we did the sickle cell. And you can see almost a gene dosing effect where in the sickle cell animals, you see the large amounts of cathepsin K in the carotid artery wall, where we also saw the elastin breaks by just 1 month. So this is no perturbation. This is no intervention study. It's just the genetic disease. And so there was a clinician on my student Philip's dissertation committee, and she said, why don't you see if you could treat the mice with the J&K inhibitor? Now, this J&K inhibitor will not be used in humans. It has a lot of cross-reactivity, and J&K is kind of important to lots of signals. But this sickle cell clinician, she said, listen, they have elevated inflammation. We need something. Let's try it. So we then injected sickle cell mice with our J&K inhibitor, and we had controls as well. Daily injections for 2 months, with the question of the systemic inhibition of J&K prevent that sickle cell mediated pathology. And again, I love all this data. It's so exciting. So you can see in the mice treated with the vehicle control, you can see this enlargement of the artery, duplication of the elastic lamina. But when we treated the mice with the J&K inhibitor, you can see the SS mice retain the same structural and physical properties as the wild type or the heterozygous control. So this is from ages 1 month to 3 months, so the earliest age groups. And then we went and quantified the elastin breaks. And again, in the vehicle control, significant amount of elastin breaks, elastic lamina fragmentation. But when we treated the mice with J&K inhibitor, we were able to block that. And again, with the cathepsin K staining, we see cathepsin K is elevated in 3 months. sickle cell mice carotid arteries, but with the J&K inhibition, we can block that being upregulated. And so it all is looking like it's going well together. But the reason we care about elastin as well is because, again, it confers elasticity to the vessel. So as the heart is pumping that blood flow out of the aorta, then the arteries expand, and then then the elastic recoil contracts back on the blood to keep the pressure wave up, to keep the blood flow moving, so that the heart doesn't have to do all the work. But as you damage the elastin, you lose that elastic recoil, and the arteries start to get blown out, and the heart has to work harder. That's the complication of atherosclerosis. So working with Rudy Gleason, we began to look at the mechanical properties of these carotid arteries in the sickle cell mice. Now, and Victor really did this study. So our hypothesis, I'm trying to get that movie to play. Come on, movie. Our hypothesis was that, not get the movie to play. So we could cannulate a carotid artery onto this pressure reactor, where we would increase the pressure and we'd watch the diameter of the artery increase. So we were doing pressure diameter curves. Our hypothesis was that with degraded elastin, we would have stiffer arteries in the sickle cell animals because you're losing the recoil. But that was not what was happening. I tell you, sickle cell will turn you upside down if you study it. And it turns out that, again, if you look at the age match, 1 month or 3 months, the sickle cell animals at 3 months had an increased compliance compared to their littermate controls, which was the heterozygous. So this puzzled us. We knew the elastin was being degraded and we weren't quite sure what was going on here. But then we carried the animals out to 5 months. So from 3 to 5 months, we did a new regimen, again, 3 to 5 months, treating the animals with the J&K inhibitor. And this actually really blew our mind. Look at these 5-month carotid arteries in SS. These expansive outward remodeling is just like crazy. And so this now actually starts to look like an aneurysmal vessel, right? An aneurysm, which is this expansion of the vessel. We quantified that the differences. There's a thinning of the medial layer. But again, the J&K inhibitor was able to stop that transformation. And then when we went and stained for collagen 1 in the artery, remember collagen 1 provides tensile strength and is more resistance to the change. The sickle cell arteries, this is a Mason trichrome stain where the blue indicates collagen 1. And you can see in the heterozygote control, you see some, but in the sickle cell animals where you have that blown out vessel, you can see that the collagen 1 in the medial layer is like almost gone. And we actually then pulverize vessels to quantify it biochemically instead of just looking at the images. And you can see that there was a significant reduction in the SS mice, but when we treated those mice with the J&K inhibitor, we could protect the amount of collagen in the vessel wall. And also you can see that from the image. So this is important because the collagen 1 does confer that tensile stiffness, and that may be why we were seeing increased compliance, because the collagen was also being lost in the sickle cell animals. And we did the mechanical testing afterwards from the 3 to 5 months. And again, the results were interesting. In the radial direction, we actually did not see protection we thought, right, from 3 to 5 months. But in the axial direction, we did see an increase in vivo strain of the SS mice treated with the J&K inhibitor that better approximated their littermate buddy controls. So the summary for that part is that cathepsin expression activity due to sickle cell, J&K can protect it. And there are these differences in the mechanical properties that are changing. So we think the elastin is being degraded first, and then later we're seeing the collagen being degraded. And it's important to know, I didn't have a slide on this, but in the young children, they're at risk for what we call ischemic stroke, where there's a blockage that causes a stroke. But then when people get out of their teenage years, the risk of stroke drops. But then in the early 30s, there's a risk of hemorrhagic stroke, which hemorrhagic is where the blood vessel breaks. And so when we look at this loss of collagen and loss of that mechanical protection here, that could be the clue to why we're seeing the hemorrhagic strokes later. Whew. Then what about hemodynamics? Oh, I'm out of time. Let me do hemodynamics really, really quickly. Okay. So again, I told you about disturbed blood flow branches, bifurcations can induce this disturbed flow that causes remodeling of proteases. Well, look at this, the internal carotid artery entering the skull and the medial cerebral artery. It's like all these twists and turns. So clearly there's a lot of potential for disturbed blood flow. So my student Christian, what we began to do is do computational fluid dynamics on children, on the arteries of children with sickle cell, so we could see, are we seeing what we hypothesized were disturbed flow? Are we actually seeing that in children? So we were able to obtain MRA scans from a clinician. And then we would use the DICOM files and put them in silico, and then Chris would do the CFD on them. And so what thing we saw, these are non-sickle and these are the sickles. And the first thought, it looks like, okay, we don't know what's different, but you do see there's a lot more bumpiness in the sickle. I'm saying that now, we did not realize that at first, and I was kind of like, what? But when we actually then, to test this, input the same fluid profile into the internal carotid artery for the non-sickle and the sickle, just to understand how does the geometry or the anatomy cause changes in the fluid flow. And these are rigid wall models, so these are not, so I know there's no fluid structure interactions. But it was crazy to see that in the sickle vessels, with the same inputs, we were getting those middle cerebral artery velocities greater than 200 centimeters per second. And this is just due to the anatomy, right? And we were not seeing that as well. We see it a little bit here, but you were definitely seeing that in the sickle. And then when we went back and looked, there were also regions of flow recirculation in the sickle arteries, but we didn't find that in the non-sickle arteries. And so we've gone back now and started to hypothesize if these bumps that we're seeing, or these micro features, might actually be some aggregations of cells or other things that locally can disturb the blood flow, and that now why we maybe have more pockets of disturbed flow throughout the cerebral arteries that we would see in a wild type individual. But we still have to, we don't have human arteries right now. So we're going to use that in our mouse model. So what about the cerebral arteries in the mice? Well, we wanted to do longitudinal studies because there's great variability in people with sickle cell and in our animals. So we developed that, well, we didn't develop, it was published, and we then optimized it for our use in our hands. This is Hannah Song, a research engineer in my group, where we did label-free MRAs so we could illuminate the arteries. So then we could follow the same mouse in time from 1, 3, and 5 months. And so this is just from 1 to 3 month data here. You can see that in the AS, the littermate controls, the heterozygous, you see 1 month, pretty homogeneous, and at 3 months, they're larger arteries, as you'd expect, pretty much together. But in our sickle animals, 1 month, kind of together, there's some variability. But look at this difference we're seeing in the 3-month SS mice, that these arteries are much larger, so the dilation is already happening. It's not statistically significant because of this huge amounts of variability. So we're collecting more of these samples to get those numbers up. And that's just for the carotid artery, not the cerebral arteries. But with my collaborator at Peking University, Georgia Tech has a graduate program with Peking University in Beijing, Emory University, and Georgia Tech. My student, Christian, went over there, and our collaborator has this way of plastinating the vessels that then you can do micro CT to really get the features of the smaller vessels. And then you can actually segment out the images that you want and isolate your particular arteries. And so Christian has done this so we can get the carotid artery, the internal carotid, the middle cerebral, and the anterior cerebral artery. And then we can reconstruct those in silico to do our computational fluid dynamics. And what he was able to see, we did this over several ages. He did 4-week, 12-week, and 24-week. And that 12-week was that sweet spot where we were seeing this increased centerline velocities in the sickle cell animals and not so in the wild or their littermate controls. So this is data we're still breaking down to, again, look at. And then what we love about the mouse model is we can now match the CFD with actual histology to start to look for these microfeatures, cellular aggregations, and start to put together what might clinically be a problem. So just to end there, the altered hemodynamics and elevated blood velocities are recapitulated in the mice, which is exciting. We've done ultrasound as well to confirm that. But there are these age-related changes to the extracellular matrix and the hemodynamics that has an effect in the sickle cell animals, which might give a clue why the young children are at risk for a certain type of stroke and older individuals are at risk for another type. And again, our goal is to match the histology, which is the biochemistry, with the CFD and the biomechanics so that we can determine these mechanisms of the cell-mediated matrix remodeling. But if you don't get anything from this talk, I want you to understand there's so much to know about sickle cell and it affects every organ system. So if you study the eyes, the kidneys, the gallbladder, bones, these red blood cells cause problems systemically, and there's room for you in the field of sickle cell research. So he stole my picture, but I love to thank my whole lab group. It was a momentous day, and these are like former high school students, undergrads, grad students, postdocs that all came back. Thank my funding, and really thank you all for inviting me to your ME seminar, and happy to have discussions. Thanks.
Manu, thank you. This was awesome. I love the energy, and the work is really inspiring. I think there's a few questions already out there. Linda has a question, and then Ming asked a question, and Ming had to leave for childcare reasons. He has to pick up his daughter. But I'll ask a question on his behalf. So I would encourage everybody else to ask questions, and you can just put in the chat or in the Q&A, I have a question, and we bring you up so that you can ask in person, and Manu can hear your voice. So keep the questions coming. Linda, do you want to go ahead with your first question?
Yeah, but Ming already left because otherwise he could trump me.
Yeah, so he has asked the first question. That's true, but he left. I can ask his question.
Okay. So let me just Manu, that was first of all a beautiful seminar. I learned so much from that. Because of course I'm super fascinated about the Jun kinase. But before I ask you about that, can you just clarify why the transfusions are helpful when there's the increase in velocity, because it's an occlusion? Is it somehow modulating inflammation, or what is it about a transfusion that helps when clinically you see that velocity? Can you close that?
So that is a really, really good question. So I don't have an answer. So the thing here's the other problem is when they see the elevated centerline velocities, they normally do not see a constriction in the MRA. That only happens way later stage. So for some reason, the velocities are elevated, and at least it's not an MRA detectable stenosis. And so that's why we've been looking at the downstream tributaries to try to understand where are things being narrowed off that's affecting the flow in the measurable arteries. So that's the other conundrum. Yeah. Great observation.
So I have two more questions, and one is another sort of easy one, and then we'll do Jun kinase. So right now I'm writing a grant actually on endometriosis, where we're going to stratify, we're going to look at groups of younger patients and older patients with their, what we call the microenvironments. So activating, like PBMC activation, like you have done so many times. And younger people are fundamentally just different in their inflammation than older people, at least in the women category. I don't know about. So when you think about the lifespan of these kids, are there things going on, like when you do those assays, which was really fascinating, have you been able to look at younger versus older? Is there anything known? Do you get enough samples in terms of inflammatory response and so on? Are there fundamentally just more aggressive things in little kids that then set up for later failure, or do you know?
So we don't know from the humans. All of our studies have been in children. So I think they were between 5 and 15 or 16 for the human samples we got. That's why we started to look at the mice, because we're seeing the age-related differences population-wise, and that's why we wanted to ask those questions. But the other thing that modulates sickle cell severity of disease, in humans especially, is the expression of fetal hemoglobin. So normally people turn it off, but in sickle cell disease, different people keep it on at different lengths of time, and the more you have, the more moderate the disease is. So we actually did not measure that in the blood that we were testing.
Got it. Okay, so then the other thing is, obviously there was just a reported failure of a new sickle cell drug recently, like this past week. There was one reported. I almost emailed you I always get these updates. But the Jun kinase inhibitors are so fascinating, because this is like what we've been thinking about for endometriosis. They also, of course, hit macrophage, not just the endothelial cell, and they really are fairly specific. And then there's there's 3 kinases. 1, 2, and 3. Apologies, I'm having terrible allergies. So I think maybe it is realistic clinically. Have you talked to pharma?
We have not, but I'm glad NIH doesn't buy it. But maybe if we talk to them together, we could talk them through it, because we have not seen early death with the mice that we treat. We don't see other complications. So I think that theirs is so ramped up that it might just bring it down to a regular level and doesn't cause problems. That's my thought. And these highly inflammatory diseases.
We're trying to get, there was a clinical trial for a Jun kinase inhibitor for endometriosis, and it failed, but for reasons I can talk to you about later. And I know a guy who still wants to get it in the clinic for endo. And I think there are clinically relevant ones. So you and I should talk offline and not.
Yeah, because I'd love to test some other J&K inhibitors. I'd love to, yeah.
Yeah, and it's really Jun kinase 3 that you need to hit. And so we can talk offline. But of course, I'm going to put in the chat the picture of us running the sickle cell road race. Right out there. If you go to Atlanta in April, Manu will make you run in the sickle cell road race with him. And I'm going to put that picture in the chat.
Linda beat me badly.
I was very proud of myself.
Thanks so much for those ideas, Linda. Thanks. We'll talk. We definitely need to talk.
Let's talk offline because I had no idea about that story.
Evelyn's here as well, our department head. I just sort of promoted her panelist if she wants to ask a question, but I asked Ming's question first. Ming asked a question that's close to his heart about what makes the mechanics of these sickle cells change, the viscosity in particular, right? Is it just the membrane, the cell membrane, or is it sort of the interaction between cell membrane and intercellular matrix and cytoskeleton, if you want?
Oh, no, I mean, the way that I've seen the reports, now we can do some additional studies if he has other questions, is that membrane damage. So there's ion channels that start to be damaged on the membrane surface due to where the polymer has started to poke. Phosphatidylserine, of course, gets flipped out, but the red cells start to dehydrate and it damages the lipids and it's membrane viscosity. So why it's super interesting, because this is why the blood becomes more viscous. And I learned this when we wrote this review paper, because normally the red cells, like as the fluid is going across it, like the red cell phospholipids just zip around. But as they become more viscous, they start to resist flow as an individual phospholipid that then is going around the whole red cell. And then multiple red cells are all resisting that flow, and it just slows the whole thing down, which is a crazy complication. There's a group at Brown that does modeling of the red blood cell membrane. It's a Greek group. I'm going to forget the last name, but I think Doug knows this person. But yeah, that's what they think it is, this membrane damage that is really driving it.
Okay, so it's not the interaction between membrane and the intercellular stuff. It's really sort of the proteins and the molecules on the membrane that change.
So it could be a little bit of both, because, you know, red cells have that special membrane scaffold, right? The spectrin, things and all that. But the way that I see it mostly talk about is viscosity of the phospholipids and the proteins in the membrane.
Okay. I think that will be something that satisfies Ming.
Okay.
He'll probably watch the second part of the recording when he's back with his daughter from daycare. And then he might catch up with you.
Sounds good.
There's a question on the J and K. When you put a J and K in, you really show that you can inhibit that expansion of the vessels tremendously, right? It's incredible. And you have done this in mice. Can you take these really cool sort of findings and put them in children, or is that something where you say, okay, this needs a couple of other things to be really sort of sorted out before that will fly?
Well, so I've gone to a few stroke meetings and other meetings, and I went to the stroke meeting when Bob Adams, the clinician who did the STOP study with the monthly blood transfusion, was being honored, and I talked to several other neurosurgeons there and was like, can you do any interventions with the children? And they're like, doing studies with children is hard because no one wants to be the one to damage the child. So they were talking about the groundbreaking nature of what Bob Adams did to even do the STOP study. And I was like, but the children, if they don't make it out of childhood, like, you know, what are we doing? So I don't know the boundaries to get it into actual children. And we probably need to do a little bit more before we get it. Maybe we need to do some other model, but because it's a genetic disease, I don't have an answer for that question, but thank you for posing it. I don't have an answer.
But what you're saying is there's a couple of intermediate steps where it has to be.
I think it's challenging to get clinical trials in children. I mean, NIH is changing that now, right? Like even in our own studies, if you do human samples for your own regular just research grants, then they want you to get samples from all age groups. So it might be changing, but I think we're not ready to just inject it into children yet. But some of the more riskier ways of dealing with sickle cell, like the CRISPRs and some of the other things, those are being tested on children that are having massive strokes early on and are just having a much more dangerous time with the disease. Then those are the ones that they can put up for some of those more riskier trials if the parents consent.
Yeah. Felipe has a question. I'll bring her up so that you can ask the question yourself. Let's see. Good work. Felipe, I just gave you panelist status and she's here.
Can you hear me?
Oh, he is here.
Yeah, well, hi, thanks, Manu, for the great seminar. I'm always smiling and I'm keeping smiling right now, so well, thank you. My main question was something to do with the reassembly of the arteries that you were recalling on the mice with the sickle cells, and maybe that reconfiguration, does it have something to do with this EPO signaling pathway and the JAP-TAS activation? I was just wondering because I read a while ago that the JAP-TAS signaling was something to do with the act as a mechanotransducer for cancer cells, so they can recognize on the low density and high density of cells, and that can interact on the transcription factor. So I was just wondering if the sickle cells might have something to do on the reassembly on the arteries. I don't know if I explained myself well, so sorry.
Well, so let me ask you, so I don't know this hippo signaling pathway, but the other one you said was what, JAK-STAT? Is that the other one?
The YAP-TAZ. Yeah, it's like the proteins that enter into
Oh,
the...
YAP-TAZ. YAP-TAZ?
Yeah, YAP-TAZ, yeah.
Okay, I have not seen that, but that doesn't mean that it's not there because I have not looked. But I will take a look. The hippo and the YAP-TAZ. I've heard about YAP-TAZ for like stem cells and other things, but I haven't looked. But I can take a look and see if that's involved.
Yeah. Oh, okay. Well, thank you. That was my only doubt. Thank you very much.
No, thank you.
Manu, there's one more. If you were a student now and you would study these pathways of mechanotransduction, the interaction between the outside of the cell and then the sort of pathway to get to the nucleus and re-expression and then back outside of the cell, what would you recommend to a student who just gets into that in terms of how on earth do you make sense of the complexity of these different sort of cascades of signaling and proteins, things like this, right? How do you go about this? Do you take a physicist's mind and you say, okay, let's just simplify this to an extent where it doesn't work anymore? Or do you say we have to be close to the biologists, learn the language, learn the sort of the insights, the quantitative sort of aspects of it, and then sort of try to put the mechanics and the physics into it?
So, okay, excellent. But so I'm biased, as you can imagine, because I took my biology background where, you learn reductive biology and you do one experiment at a time and blah, blah, blah. And then came up to MIT to work with Lyndon Doug to do systems biology and look at the whole system, right? So I think someone starting now, there are some great system biology tools that person who should start appreciates, as you mentioned, the complexity of the system, and then not think that they can reduce it to one thing because that'll be the engineering way to simplify it, right? I think it's about appreciating what is known, appreciating the complexity, recognizing that biologists have certain ways of asking those questions, but I think also recognizing that computational approaches help understand things that cannot directly be tested for experimentally, because then that's how we kind of fill the gaps and then generate other hypotheses to then show it the right way. So I think you need to stay close to both. And I think it's a risky thing to be the arrogant engineer or the arrogant biologist and think that you have the way, because the one that kind of dances in between, I think will, have the best score. That's what I would say, but recognize it's complex.
Thank you.
Yeah.
Okay.
But come on over, mechanical engineers, come on over. It's so cool because it applies to like astronauts and microgravity. Like that's how I kind of got into it, and just so many different things. It's really just an amazing way that our body responds to mechanics. I mean, it's amazing.
But you heard Manu, come on over. I think that's
I think Manu has a way of pulling people in, and I especially have to say, given that you were trained in biology, the way that you think has always been an engineering way, a systems way, from a molecular up to system scale, and the ability to put teams together to go from the clinic down to the basic molecular science is truly a remarkable thing. And I really, as you know, I really admire the way that you were able to go from the clinic down into the molecular pathways. So congrats on an awesome talk. I really enjoyed that.
Thank you so much.
I'm writing you about June kinase right now.
Okay, I look forward.
Okay. Thank you. Awesome talk.
Later.
If anyone wants to hang out with Manu a little bit more, I can promote you to panelists or whatever you want. You can ask him questions, but I think that's a good point to formally close the seminar. Manu, thanks a lot for speaking to us today. This was really cool.
Thank you for having me. This was fun. Thank you.
Good to see you.
You too, Linda. I'll be in touch.
Are you in Atlanta right now?
I'm in Atlanta, not going anywhere.
Yeah, no, none of us are.
Manu.
Thank you, Don. Thanks a lot. Just making sure my mom stays put in New Jersey. She's doing okay, though.
Is she? Good.
She is, yeah.
Yeah.
All right.
All right. Ciao. Bye.
Okay. Thanks a lot.
Thank you.
Someone else hanging out, oh, there's another person.
Yeah. Gomu has some question now. I'll promote him to panelist. Let's see what he says.
Oh, I'm sorry. I was typing a question and then I got, wait.
And Andrea has a question as well.
Oh, sorry.
Sure.
Let me start my video.
Thank you, Andrea.
Hey, sorry.
Hello.
I was typing a question and then the chat went off. And I was like, oh no.
Oh, Marisha.
No, great talk. I really enjoyed it. I have my background is in biomedical engineering, but I'm getting my PhD in mechanical engineering now. So I really, really enjoyed your talk. Hey, Becca.
Thank you.
Yeah, I just wanted to ask, is it known, you mentioned that the sickle cell condition impacts basically every organ system. Is it known, or do you have a sort of hypothesis about which organ systems are impacted first? And of the different organ systems, are there different timescales sort of governing how rapidly they experience negative impacts from the condition?
Oh, that is interesting. I have not seen the breakdown like per organ, but the main cause of death is acute chest syndrome. So the lung damage is because there's get scarring and fibrosis in the lungs. And again, the lungs are so critical to getting oxygen, CO2, and you don't have that, then you die. That is the most common cause of death if people make it out of that early stage. They get bone, they need hip replacements. That's the one we're talking about, but there's a high percentage that need hip replacements by late teens because the femoral head of the bone just gets worn down and there's increased osteopenia. Then the retinas are more later stage in life, but the retinas also, they start to get damaged even in people with sickle trait, where older people with that get problems. Kidneys start to fail later, but the kidneys, I think, they would fail after some of the other critical things are problems. So I think they have a longer time scale because I think the filtration there, like the blood flow is different. Like, you know, they got the huge tubules. This is my own conjecture. The huge tubules is not the smaller things. And I think the sickle cells would never leave the glomerulus. So they don't, you know, so I think but I do, as I talk to you about it, I think there might be some staging of when it becomes clinically a problem. But the lungs and the brains are the big ones that want to protect those vasculature first off.
Okay, thank you.
Yeah, that's a good question. I never thought about it. That's good.
I had a question, but I'm a biologist, so this is not about me.
Okay.
So
There's
no,
a superstar biologist on your lab. Okay.
So I was watching, listening to your talk, and I was very curious about the role the leukocytes play, and I just thought of the leukocytes as kind of just being thuggish and grabbing onto a sickled cell, not letting it go, and messing up the flow essentially. Yeah. But I'm not a mechanical.
Even worse. So this is the part I didn't mention. I probably should have mentioned it. So people with sickle cell also have monocytosis. So there's increased numbers of circulating white monocytes in their blood because of the chronic inflammation. So not only do they grab onto things, there's more of them. They are more adhesive, and then when they stick to the wall, they turn on proteases that start to wreck shop. So yes. Yeah. But the other problem that they do is because remember, leukocytes have a nucleus. Yep. So when that sickle accumulation starts, if the red cells stick to the wall first, then they tether to a white blood cell. Now that's got a rigid nucleus. So that's like blocking things off more so than just red cells alone, because then the nucleus is like, well, we can't deform. We're not deforming. And so all of that, yeah, is a problem. So then they do play a part.
Yeah, Nara was like, what's your question? I said, this is not my talk. He's talking about mechanics. I have questions about biology.
That's great. I like to do both. You know that.
So then, with regard to the leukocytes, is there a way to address the leukocyte challenge to let's say alleviate some of the stress in the flow?
So the way that people want to do it, so the one drug that's on the market now is hydroxyurea. And what does it do? It actually promotes fetal hemoglobin expression in the red cells. That stops the sickle polymer from forming fully and deforming the red cells. And that then stops all of the other complications or reduces all of the other complications. That is, the best bet is if you can stop the cells from sickling. Now, for other people, but then some people don't want to use it because it's an old cancer drug. And, Black families are like, what you trying to get my baby?
You trying to get me cancer?
That's another story. But the other issue with leukocytes that people are using, one of the drugs, I got to find out which one Linda said just failed, is it actually binds to selectins. It's an antibody drug that binds to selectins on the endothelial cell surface so that then the white blood cells or the red cells don't attach and they just keep flowing. But again, a drug like that, you've got to, keep a lot of it in there because, you're coating the whole system. But that one was approved, I think, last year. But I think it's about how do you stop the damage? Then you won't need all of the white blood cells to be produced to fight the damage. That's, I think, is the hypothesis.
A lot of ideas, but it starts with anti-racism. So I'm going to just leave that.
Good to see you, Marisha. Good to see you. And this little one too.
Yes. All right. Well, thank you so much. Don't want to keep you.
All right. We'll be in touch. Thanks.
Thank you so much. This is a great talk. Thank you for sharing.
Happy Friday.
All right. Ciao.
All right. Did we lose Ngomu? No, we lost
I'm here. Sorry, Manu, that is a nice talk. I need to keep on the mask because I'm in the lab with other people.
I'm in Georgia. Keep the mask on. It's great.
Yeah, yeah, so it was a brilliant talk. Actually, I did my first postdoc at Emory University School of Medicine. Now
Nice.
I'm just down in the School of Bioengineering here at I'm a postdoc in School of Bioengineering here at MIT. So it was a brilliant talk. I was more thinking about your computational approaches. And I was thinking, since the model is a little bit complicated, is it possible to use macrofluidic design chips to do those, to start those artery? Because with a macrofluidic approach, you can control both the size of the artery. You can control the pressure. You can control the amount of red blood cell you flow in or other cell you want.
But what did you just call it? Microfluidic, right? So there's lots of work in sickle cell in the microcirculation where people are using microfluidic chips and the rest, but it's the large arteries where you're seeing this remodeling and the strokes. And so as you know, the flow profiles can be much different in a large artery compared to where you start to restrict to a microfluidic, depending on, you know, how many cell diameters across are you doing, that you won't be able to get some of the more complicated flow profiles that we see in the large arteries. So that's why we that's why we don't think, again, others are using those approaches because again, you bring up all the good points that people can control and then watch and visualize. But because we want it to be able to be the more complicated fluid profiles, that's why we look in the larger tubes. So that's why we don't use the microfluidics for our studies directly.
Okay.
That makes sense?
Yeah, that makes sense.
Yeah, but there's a lot of other good work because again, the microcirculation has lots of complications. That's where the crises occur and all that. But I'll tell you some interesting facts then too. Nobody asked me this one. So when I first started studying, there's been some great biophysical studies that were done at NIH like in the 50s by this guy named William Eaton who just published something amazing like a month ago, where they showed that the time it takes, because some people ask me, well, if you're doing large arteries, then that's oxygenated blood, then you probably don't even have any sickled red blood cells in there. So what's happening, right? But it actually, his studies from the 50s showed that the time it takes for that sickled polymer to melt after the red blood cell takes on oxygen again is actually much longer, not much, but is longer than the time it takes the red blood cell to traverse throughout the circulation, right? So it can still, after it gets oxygen from the lungs, can still be sickled as the polymer is starting to the polymer is melting, but you can still have this sickled red cell floating in the oxygenated circulation. So there's all these other kind of tricks that are going on because the reason we care about it being sickled in the oxygenated blood, and maybe Matthias or anyone else has a comment here, we think that the red cells become more dense, and if you have way more dense cells, are they then able to break streamlines? Because we normally don't think of red blood cells as breaking streamlines because they're neutrally buoyant with the rest of the blood flow, but that's because they normally are. But as a red cell gets sickled and stiff, density goes up. Also, as the red cells age, their density goes up. And then there's a question of how many does it need to break a streamline when you're coming around those tortuous curves in the cerebral arteries. I don't know, but I keep trying to find fluids people who want to model like rigid bodies within a fluid flow, and like everyone's like, whoa. That is a complicated problem. And I believe it, that's why I haven't done it. But that's why I think then we need the larger vessels outside of microfluidics, because you see some of these vortices that happen and they just wouldn't happen in a microfluidic system.
There is a guy here, really junior.
That is a very good argument. Working with a macrofluidic for a while, I don't think there is a reason why people should keep the size of the chips on the macrofluidic skull. There is no reason at all. It's just because of most of the initial experiment in macrofluidics, they did not matter about the size.
Then you get macrofluidics. I couldn't call it microfluidics if I was in like
Yeah,
the millimeter
but
scale. They'd be like, that's millifluidics. And I'd be like, yeah.
yeah, so that is also possible. You can have a chips with that. It's just you'll have less things on it, but it's going to be a bigger chip. You can still use the same material.
That's something I'm interested in. That is something I'm interested in.
Yeah.
If you think we could do one at the scale that we're thinking about, because I even worry that with our mice arteries, that they actually are still much smaller than the human, and we might not be seeing some of the profiles, but I think the mouse is still, it's physiologic, it's a whole mouse, right?
I think you can do up the size of the what is the part for transfusion that we use in the clinic for transfusion. Up to that size could still be read on a good microscope and monitor by a computer.
There you go. And so when you start your own lab, my friend, we collaborate. But no, thanks for the suggestion. Thank you. Anyone else?
Yeah, I have a question. Thank you for the great talk.
Thank you. Thank you.
I really love the scales from studying like patients in the clinic and as well as going all the way down to the molecular scale. So my question is, what do you want to do next?
Okay, so again, we want to get a drug that is actually going to make it into the market. We would love to do that, right? But the immediate thing, we've got a grant that's been under review, who knows what they're going to say about it, is we now, because we see these age-related differences, it's really interesting. I have not before seen reported these blown out arteries in people with sickle cell in humans, because I think they stopped doing autopsies on them. Sometimes the families don't want it to, and it's also Black families who like, don't cut up my baby. You know, it's just all these cultural parts that go with it. But what we really want to do is we wonder, because the question is, if once that cerebral vasculature is damaged, it stays damaged. Our questions are, at what point can you stop the damage and retain and prevent like stroke damage, right? So again, you saw we did where we started at 1 month, then you saw we started at 3 months. We have proposal studies where we're starting at 5 months, because some people just don't start engaging with the clinic. They just deal with their problems until much later, right? And we think already that what we've seen, that the mice that we started at 1 month do way better than mice started at 3 months, right? As far as vascular damage. But we actually have not done a direct comparison of a 4 month study comparing a 1 to 5 month versus 3 to 5 month. But we also want to go out even further than that. Then what we also have is we made a transgenic mouse now. It's a sickle cell cathepsin K knockout mouse. So it has sickle cell, but it does not express cathepsin K. And so we're hoping that that then will show us the particular role of cathepsin K directly in the vascular damage that we're seeing. But what we like about that is we can also do bone marrow transplants to see which cell type producing cathepsin K is the more important one. So then if it's like monocyte macrophages, well, then you can use like liposomes and other things to target the monocytes to, you know, tell them to stop doing things. So we're trying to tease that apart to directly show the role. The reason why, again, the cathepsin inhibitors are not in the medicine cabinet, right? And so, because we would like to directly target those. We only target J&K because we can't directly target the cysteine proteases. So those are the things we're thinking about as far as protecting the brain and the vasculature. And also our other question is, again, the only curative functional cure for sickle cell is bone marrow transplants, because you have to get rid of the hematopoietic stem cells. But it has been shown in individuals that even though they've had a bone marrow transplant, they still are at risk for some of the cardiovascular damage and problems. So again, it's like the vasculature is damaged, but even if there's way less or no more of the sickling red blood cells circulating, the complications are exacerbated. So we want to kind of use our mouse model to determine like what are some of these causes there. And then if our protective strategies, like if our proteases are just turned up and they stay up, then do you still need to inhibit them, even though a person has had a curative bone marrow transplant? Or are there other ways that you need to continue to protect them from getting strokes, even though they themselves are not producing the number of sickle red cells? So we got a lot of different directions. Plus we're doing a lot with the bone also. I have some great bone work that we've been doing because cathepsin K is involved in bone degradation and all that stuff. So those are kind of where we're headed.
Awesome, thank you. Do you think there's any use in monitoring the patients in their home to prevent some of the symptoms, or like when they should go in for a transfusion, if that's the current standard of care? No,
well, the current standard of care is if it's high, then it's a month. It should be a monthly blood transfusion. And the thing is, the patients normally stop going around late teens. The clinicians would rather they continue to get monthly blood transfusions, right? But the risk drops off in your early teens, but there's still a risk, right? So that's, that's the that's the other part. And then there's, it's just, sorry,
Great, thank you.
Yeah, you're welcome.
Quick question, great talk. I really like how you went from biology to engineering and kind of sitting in the middle. I started in a bio lab in high school and kind of said that wasn't my tea, and then went to mechanical engineering and then finished my PhD. Now I'm going back to bio things now.
There you
it's
go.
Kind of funny. So I guess so I mostly work in like imaging side of things, ultrasound and some optics as well.
Okay.
I'm curious as to like, is there something that's like kind of the holy grail or something that you would like to see that is currently unable to see in terms of the in vivo models or even some of the cell models?
Yes, so many things. When we do that plastination, we have to sacrifice the animal, right? Because like the body's exhibit, that's what I call it. We are supposed to be starting a collaboration with a group of Cornell that can do capillary imaging at the surface of the brain. We're trying to think about doing light sheet microscopy. I mean, the point is, where are there constrictions in the cerebral vasculature that we can pick up? That's our big question. That might be changing the whole fluid profile that's causing these accelerated centerline velocities or causing something else. And there's just so many, I showed that one picture with the image of the plastinated vessels. There's so many tubes, right? Or is there an ordered way to do the analysis there of which ones are changing shape? We did, in other studies, we looked at the reduction in the diameter along the length of the vessel. We looked at the we've tried to all these other, not a lot of ways. I mean, it's just a little bit with undergrads over the summer. But what are we missing? What can we not see? You asked what do we want to see? We want to see where you might see a statistically significant stenosis that might be regularly picked up that's causing a problem. But we don't, right now we don't know at what level of the vessel it is. We would hope that you would see it in a major vessel and then you say, look, this is stenosis, it's a problem. But that's not what's happening. Also, I'd love to see red cells or white blood cells that are stuck to the wall live in the mouse without having to do like a cranial chamber with fluorescent tag cells or something like that. Like, where can you see it? Because the thing with these strokes in children with sickle cell is we call them casually spontaneous, but they're not spontaneous. But you don't know who's going to get them. 11% get them, right? So why that 11%? And that means also we don't know when they're going to happen or where. And so in our mice, sometimes the mice just drop dead. And if we don't get to the mouse body before the veterinarians do to freeze it, then the brain is mush within 30 minutes after it's dead. So we still can't collect it and then, do processing. So how do you see things without having to open the mouse? Or how do you see all parts of it without having to look individually with rigor? And, those brains, you can only make so many slices and those brains are only so big. So lots of things, So many good questions in sickle cell though. So many good questions.
Thank you very much.
If you have any ideas, email me, let me know. Okay, seriously.
Will do.
All right. Any final questions, Matthias? What you got?
I have thoughts. I think for the modeling, when you mentioned that you would love to model sort of flows around complex heart systems, I thought of a faculty that joined us about a year ago, maybe 1.5, Wim van Ries. He worked with Maha, who's now at Harvard, was at MIT.
Spell the last name for me.
Van Ries, V-A-N and then R-E-E-S, Wim.
Okay.
Wilhelm. He's Dutch, does some really cool fluid mechanics, is in the ocean engineering branch, but looks at vortices and turbulence and stuff like this. So it sounds exactly what you would want to look at sort of breaking of flow lines, things around sharp, hard corners. Right? I mean, I can write him an email and say, hey, there might be something interesting that you would never have thought of that Manu has here.
That would be great. Please do that. Please.
The guy is awesome. He's really, I think he's fun to work with. And then the other thing, when he said the imaging, when Xiang asked the question, Peter, who couldn't join us, Peter So, he's teaching in the time that the seminar falls, unfortunately. But Peter has this amazing phase reflection microscopy. We use it to cut open butterflies and look at living butterflies as to how the cuticle forms. And
wait, what kind of microscope did you call it? What is
it's a speckle elimination differential interference phase contrast microscope, I think.
Oh, okay. I know Peter So from EVIX. Okay.
Yeah, you guys probably have a really good connection already, and so that imaging is pretty amazing. You can do it on living organisms, and in the brain of the mouse, you probably would have to get a bit of skull away. But then the phase reflection microscopy gives us a lot of information on really subtle sort of changes. And Peter has looked at red blood cells, not the sickle ones, but he's looked at membrane oscillations. They can resolve nanometer resolutions.
Wow, that's cool. That's cool. Okay.
That phase reflection microscopy is a pretty neat thing. Saeed Yaqub worked on this, one of Peter's advanced research scientists, I would say. And I think that could be a neat little toy to at least try to see if you can see something with it.
That is cool. That sounds cool. And actually, again, I told you one of my former students is up there working with Paula. Maybe I need to send a mouse if she wants over there.
Yeah.
Amusingly, Becca and I work with Peter on that specific system that you're mentioning to look at, seeing how we can integrate the acoustic perturbations to detect some of the underlying phenomenology in the red blood cells or other cells, or even up to the tissue scale. So it's kind of integrating ultrasound and phase microscopy.
All right, I wrote your name down.
Small world.
Peter.
I think that system is definitely worth a try. It worked for us like a charm for the butterflies.
That sounds cool. That sounds
a good system.
Yeah, there's also a similar system that uses similar technology that might be applicable that both Sean and I work on with Peter.
Oh, okay. Okay.
Yeah.
Very cool.
Good.
Sounds good. All right. Thank you. Really, this has been great. And thanks for all this after feedback. I love that. This has been a lot of fun. It's a lot of fun. Think about sickle cell disease. So many questions.
I had a student writing, and she is not in the field of sickle cells or bioengineering for that matter, or imaging. She does sort of polymer work and building sort of photonic structures. She was writing, that was the most energetic talk she has ever seen.
It's fun to relive the data and the moments. I mean, it's like, it was a wild, crazy idea. And people, you were like, oh, that's not going to work. Oh, you're wrong. And then when the hypotheses started being supported, it's just like, it gets crazier and crazier. It's so cool. So it's an exciting ride. Yeah.
Manu, can we have recorded this talk, and I'll share the recording with you as soon as it's available. Can we put this on the Mackie YouTube channel, or would you like to have this under wraps and not go beyond?
No, I think if you cut the Q&A where I told all my business and all the things I'm really working on, I think most of what I present, there's some that's unpublished, but I think that I presented it publicly before. So no, for sure, send me a link to it. That'll be great.
Yeah, and then you can take a look what stuff we should cut out. If there's a slide that you didn't want us to see, or anybody else to see, us obviously wanted us to see.
No, that'll be great. That'll be great.
But then I'll give you first tips, and then if you say, okay, this is good to go, then we can put it on the website. There's a couple of people, Peter, for example, but also Ming and Colin Bouye, who asked if they can see your talk because they couldn't come or could only come to half of it today.
Okay, no, for sure. Yeah, feel free to share. That's fine.
Cool. All right. Thank you.
All right. Thanks again. You all have a great weekend. Wear a mask. Wash your hands. COVID is real.
Well, at some point we have to get you over here for dinner and things in person. That would be.
I know. That's the part that I miss. And actually, you know what else I miss about it is I didn't realize it. Those were also like mini vacations. Like now here, like I've been working all day. I do this, then I like work some more. But it used to be I would get away from my lab, from my life, and do it. And I realized there's lots of vacation time that I've been missing without the traveling for talks, you know. So I need to just take the whole day off when I give a talk sometime.
So if I still have the rule of the Mackie colloquium, then the moment we can travel again, I just say you come to us and give the talk again. There's a lot of people that should have seen it that didn't come today. They
Obviously that works out. All right, you all have a good weekend. Thanks again.
Thank you, sir.
Thank you.
Bye.